Horacio is our soybean pathologist, and we thank you for being on, Horacio. He's really willing to come out and speak to the county, and we all really appreciate him and all of his work. So go ahead.
Thank you, Taylor, and thank you, the organization committee, for having me here today. Good afternoon, everyone. My name is Horacio Lopez-Nicora. I'm an assistant professor in the Department of Plant Pathology, and I'm going to cover some soybean disease management. Like Pierce mentioned, I'll try to cover the ones that I have been working with last season. So the agenda for today that I have prepared is to talk about the soybean cyst nematode and some of the work that we have been doing. Stay in that root area, the rhizosphere, and talk about other soil-borne pathogens. I'm very glad that Pierce talked about Fusarium graminearum, because we'll also cover that in some of the soil-borne pathogens causing seedling disease. And then we're going to jump and talk a little bit about the foliar fungicide trials that I'm conducting on a very healthy season last year, and we'll see what type of results I have from that. So to understand how soybean cyst nematode damage, I'll cover the life cycle of the nematode. It overwinters on eggs, and normally these eggs will stay in the soil for a long period of time. And we want to quantify the numbers of eggs per 100 cc of soil. That's half a cup of soil. Most of the time, we'll send our soil to a nematology lab, and we will receive numbers of cysts per 100 cc of soil. I will encourage you to request numbers of eggs per 100 cc of soil, since this is the initial inoculum. From the eggs, the nematode will hatch and will use this stylet to penetrate the root. It will penetrate any root, resistant or susceptible root, corn root, any root. But the damage will happen when the nematode establish an interaction with the plant, in this case, soybean, which is the main agronomical host. The nematode will establish feeding sites and stop moving, will continue developing, really robbing the nutrients from the plant, and this is exactly how the nematode will cause yield reduction. The cells around the head of the nematode resemble transfer cells, which we can only find on the seeds, on the embryo of the plant. So the entire plant is thinking that it's feeding the seeds, where actually it's feeding this pathogen. The males regain these vermiform conditions, leave the root and fertilize the female, and then the females lay on average 250 eggs. And this can happen 3 to 6 times depending on the conditions of the season. So you can imagine how fast the population of these nematodes can really explode in one season. So the symptoms that we normally will see, if we see any symptoms, are mainly related to stressful conditions. So we can see, when there's a drought year, we will see plants wilting more in areas with high levels of soybean cyst nematode. When there are other pathogens, such as the fungus that causes SDS, for example, in the presence of the nematode, we will see more SDS symptoms. Let's say nutrient deficiency. We will see marginal chlorosis associated with potassium deficiency. And this will happen more severely when we have more soybean cyst nematode. We will go ahead and take a soil sample, send it to a soil lab. Results will come back that we have indeed potassium deficiency. We will fix that. Plants will look healthy the next season. However, we did not took a subsample, send it to the nematology lab, and figure out that we also had soybean cyst nematode. So we fixed the nutrient deficiency problem. Plants are looking healthy. Populations of soybean cyst nematode continue to increase. If we compare resistant and susceptible, sometimes we will see stuntingness, so a decrease in the height or development of the plant. But the nematode in the Midwest has really adapted to reproduce on soybean, and most of the time we do not see visible symptoms. The roots, however, will not lie. If we go there with a shovel and we carefully remove the soil and look at the root system, with our naked eyes, we will be able to see females attached to the root. They're lemon-shaped, yellow, or very clear in color when they are young, and then they will turn darker brown in color. They are very, very small, smaller than nodules. I'm showing here a nodule. That's the bacteria that will associate with the plant to fix nitrogen. So that's really good. And that is way, way larger and bigger than the actual soybean cyst nematode females that are attached to the root. Yield reduction is very severe when we have high levels of soybean cyst nematode. It's all about these games of numbers. The more soybean cyst nematode we have in the field, the more yield we will lose. And I'll show you some data that we have produced in 2022. But between 15 to 50 or more percent of yield reduction can be observed. We can also see some symptomology when we compare susceptible and resistant. But the nightmare of the north central region where we grow soybean is that our population of the nematode have adapted to really continue causing yield reduction with absolutely no visible symptoms. So we have our susceptible lines here and our resistant lines here, and we go to the field and we just take all type of measurements to see if we can distinguish between resistant and susceptible, or between presence or absence of the nematode. So we take height measurement, branching, numbers of pods, intensity of the color of the foliage, and we just don't see significant difference until we go and we harvest. And on average, we see 30, 37% yield reduction. To prove ourselves that this is happening and continues to happen, in 2021, in one of our research stations, we threw a grid and we sample each of these 100 quadrants and collected soil samples at planting. We detected areas with high levels of soybean cyst nematode in these red quadrants. So we have 1,000, 2,000, more than 3,000 eggs per 100 cc of soil of soybean cyst nematode, and areas where we did not detect at all soybean cyst nematode. This map shows exactly the type of animal we're trying to beat. It's aggregated in space. That's the way that soil-borne pathogens work. They live in clusters. So if we have parked here our truck and took a soil sample for soybean cyst nematode, and we just sample this area and send that to the nematology lab, we would have received a result that says non-detected. On the other hand, if we would have parked our truck here and took a sample here in this part of the field, we would have very high numbers. This indicates that really when we're dealing with soybean cyst nematode, we need to do an intensive sampling across the field and the area that we are assessing. Very important. From each of these quadrants, we also collected yield and all the measurements that I talk about: height, intensity of the color of the foliage of the soybean, and these were all planted to the same variety. So we did not see any areas that differ that could tell us that we had more or less soybean cyst nematode. No visible symptoms. I'm using a box plot to show the results here. I like box plots because the wider the box, the more variable our data set, whereas the narrower, the tighter the box, the less variability in our data set. We have a very dark line here that indicates the median of our data set. And these whiskers, that's the name that they receive, whiskers, shows the minimum and the maximum value of our data set. I'm grouping all those blue quadrants here, and I'm labeling non-detected, where we did not detect soybean cyst nematode. And here will be the red quadrants, where we had soybean cyst nematode above 1,000 X per 100 cc of soil. So we have quadrants with 2,000, 3,000, 4,000 X per 100 cc of soil. On the y-axis, we have yield in bushels per acre. So we're, with a very simple math here, we can again verify that indeed, with absolutely no visible symptoms, on average, we were losing 37% yield in the presence of soybean cyst nematode above damage threshold. With the price and being a bit conservative with the price of soybean in 2021, we're leaving $300 per acre with absolutely no visible symptoms. We have no idea that we are losing $300 per acre on average in this field. So what do we do? What do we do if we have a problem that we cannot see, that there is no symptoms that can tell us that we have it? Well, soil testing is the first one. An active management of soybean cyst nematode starts with knowing if you have the problem. And you're gonna know if you have the problem if you take a soil sample and test for soybean cyst nematode. If you do, you wanna know your numbers. Do you have a legal soybean cyst nematode and the amount is one that you could keep on check? Or do you have above damage threshold that you really need to integrate different management strategies? So NIBLAC in 2005 recommended these 3 R's to manage soybean cyst nematode: rotate, rotate, and rotate. Very simple to remember. The first rotate is to rotate crop. If we plant something that the nematode cannot reproduce, we will reduce the population that season. So corn, small grains, sunflowers, anything that the nematode cannot reproduce on. Rotate soybean varieties with resistance to soybean cyst nematode. We don't want to train the nematode to eat and feed on the same genetic material. Unfortunately, more than 90% of the commercially available resistant soybean varieties have these PI 88788. Nonetheless, rotating those varieties is recommended to manage soybean cyst nematode. The third rotate is to rotate sources of resistance. So I just mentioned that PI 88788 is the most common one, but there are others. Peking, for example. So find varieties with a different source of resistance and rotate those sources of resistance. SCN is best managed using resistance when we don't want to rotate to a different crop. Unfortunately, SCN continues to adapt and reproduce on PI 88788, which is present in more than 90% of the commercially available soybean varieties with resistance to soybean cyst nematode. Now, what does it mean it continues to adapt and reproduce? Well, in 1954, we discovered soybean cyst nematode in the United States. Soon after, in all areas where soybean was produced, the nematode was reducing yield. Plant pathologists, agronomists, plant breeders got together, screened several germplasms and lines to try and find resistance to soybean cyst nematode, and they did. Several, multiple. We used 7 different sources of resistance in our greenhouse to screen for populations virulence. Well, PI 88788 was extremely good at avoiding that population of the nematode to reproduce, and it was extremely easy to integrate in high-yielding material. Soybeans that we like to grow, we knew how to grow, and yielded very well. And because of that, we successfully managed that population and used the same source of resistance for over 30 years. And you know what happened when we use the same active ingredients to manage a pest for so many years? We exert a selection pressure on that population and we shifted the virulence. We shifted the virulence of SCN that now it can reproduce on PI 88 above 10% compared to a susceptible. In Ohio, for example, we have populations that can reproduce at 20, 40, even 80% compared to a susceptible. And what does that mean? Well, if I have a population that can reproduce at 80% compared to a susceptible, that means that that cultivar that has PI 88 with that population of soybean cyst nematode, it's only 20% effective. And that's a big problem. We can identify these populations in the greenhouse. We can put populations of soybean cyst nematode, and we can test different lines that have different sources of resistance, or we can test different varieties. And I'm going to talk about a project that we are working with Dr. Laura Lindsay. So what we do is in pasteurized soil with no other organisms, we use a population of soybean cyst nematode. We release those eggs in each of those holes. We plant the different cultivars or the different sources of resistance. We allow the nematode to reproduce for 1 month in the greenhouse under constant temperature. And then we pull those roots and we dislodge the females. And then we count the females on those roots. Susceptible roots will allow the nematode to reproduce and develop, whereas resistant roots will kill those cells around the head of the now sedentary nematode and kill the nematode or starve that nematode to death. That's how resistance works for the nematode. Now we can use this to screen for resistance. So we have a system where we have soybean cultivars, we have a susceptible check, we have a population of nematode that is very common in Ohio. These soybean cultivars can be labeled resistant to soybean cyst nematode, to race 3, or moderately resistant to race 14. And we can, after 1 month, count how many females develop on this variety and compare it to a susceptible check. With that, we generate what we call a female index. And this is the project that we're working with Laura Lindsay and Connor Johnson, a master's student in my lab. We can then screen for resistance, different cultivars, different varieties that are commercially available to growers, and we can classify them. If the female index is less than 10, we'll call that highly resistant. If it's between 10 and 30, moderately resistant. If it's above 60, we're going to call that susceptible. And we did this with hundreds of varieties that are commercially available to growers. Now, the ones that I'm showing you here were labeled resistant to soybean cyst nematode. Only 13 out of the more than 100 were highly resistant. More than 54% were moderately susceptible to susceptible. And the reason we're seeing this is it's not because these soybean varieties do not have resistance to soybean cyst nematode, but because the resistance, the source of resistance that they have been overcome by the population of soybean cyst nematode we have in Ohio, specifically the one that we use for this screening. So, and that brings again to the idea of managing soybean cyst nematode has to be thought of as an integrated approach. We need to integrate resistance, yes, but also protect the seed. Use nematode protectant seed treatment. Another thing that we are evaluating in our lab, Ali Peart, a master student, is evaluating cover crops. Does cover crop have any effect on reducing soybean cyst nematode population in a field? Does it have an effect on, and this is something she will be testing in the greenhouse, attracting the nematode to penetrate those roots, but do not allow the nematode to reproduce? So it serves as a trap, a crop trap. That's the name that we give to this type of crops. And we can use this in an agricultural setting to reduce the levels of soybean cyst nematode. But what I want to cover now is one of the trials that we conducted last year. Many seed treatments are labeled that they can or have an effect on nematodes. One of those is Ilivo. Ilivo is a fungicide, fluopyrin. It works extremely well to manage the fungus that causes sudden death syndrome, SDS, but it's also labeled that has effect on protecting soybeans against soybean cyst nematodes and other nematodes. So with help of Eric Rieger, field specialist, now in farm management, and John Schoenhold, pioneer agronomist, they were able to retrieve different cultivars that I was able to use in a field trial. We did this in a research station, North Central in Sandusky. We had cultivars with that PI 88788 source of resistance, the very common source of resistance, but we also have cultivars with a different source of resistance, Peking. Half of those seeds were treated with Ilivo and half of those without Ilivo. We put this in a field with a very high initial population of soybeans nematode, one that we know will significantly reduce yield, 13,000 eggs per 100 cc of soil. We determined the virulence profile of that population is an AG type 2, which means that by definition, it can reproduce on PI 88788 above 10% compared to a susceptible. We collected soil samples at planting to obtain the initial population of the nematode and at harvest to obtain the final population of the nematode. With these two data sets, we created a new variable that is called the reproduction factor of the nematode. So it's a ratio between the final population divided the initial population. If we divide the same number, 3 divided by 3 is 1, 2 divided by 2 is 1, 2,000 divided by 2,000 is 1. If the population of the nematode is the same at harvest than it was at planting, nothing changed. The reproduction factor is 1. If that reproduction factor is above 1, that means that at the end of the season, the nematode increased its population. It was able to reproduce on soybean, on that soybean, which means that whatever we did not work. A reproduction factor of 5 means that the nematode increased 5 times more its population than what we started with. If the reproduction factor is below 1, as you can imagine, that means that something that we did in that season worked and we reduced the population at the end of the season. We also obtain yield in bushels per acre. In this case, I'm going to show. So I'm using box plots again, adding a black dot, and that's the average inside each of the boxes. I'm splitting the data set here into the ones that had Peking source of resistance and the ones that had PI 88788 source of resistance, treated and not treated with Ilivo. And on the y-axis, I'm showing the reproduction factor, and I added that dashed red line for reproduction factor of 1 just to have a reference. So results here is that on average, plants with Peking source of resistance on average reduce the population of soybean cyst nematode at the end of the season. Whereas those with PI 88788 on average allowed for a reproduction factor above 1 at the end of the season. Was not able to manage or reduce the increase of soybean cyst nematode in that field. We statistically did not detect a significant decrease in population due to seed treatment for this field trial. And then when we compare yield, which we are also interested, of course, we know that it will be not fair to compare across varieties because they have different genetic background, but we can compare treated and not treated with Illivo. And what we notice is that even though the seed treatment statistically did not reduce the population of soybean cyst nematode in the field, it statistically increased yield when treated between 2.6 and 3.5 bushels per acre. And this is great. And this is the idea of that integrated management approach. We're using as efficient resistant or source of resistance, in this case, Peking, to reduce the population of soybean cyst nematode. But at the same time, we're protecting the root and the health of the root of the soybean to increase yield overall. But this is really the data that I want to share with you. On the x-axis, I'm showing you the increase in nematode population. As I move from left to right, I'm showing you an increase in soybean cyst nematode population in that field. On the y-axis, I'm showing you yield, and then I'm splitting here between treated and not treated. It's not that important. What I want you to pay attention is that I'm color coding the cultivars with PI 88788 on blue, and the ones with Peking as source of resistance on red. And I hope you can see what I'm seeing, which is that as we have more nematodes in the field, PI 88788 source of resistance reduce its yield. The more nematodes I have, significantly, I will reduce yield on those PI 88788 cultivars. On the other hand, the Peking source of resistant cultivars are completely indifferent of how much nematode I can have in that field and remain essentially flat. I want to invite you. We're gonna be covering and talking more about this research and data set with Dr. Nick Tensley on March 7th. This is a talk that we're gonna offer with support of Ohio Soybean Council. It's gonna talk a little bit about more of the biology and management of soybean cyst nematode and some of the products that BASF is also working, not only for nematode, but for that integrated management approach and soybean health. So I invite you to register and come to this webinar. Okay, with that, I wanna just jump and kind of summarize a little bit about our survey that we continue doing throughout Ohio. We're doing this with funding from Ohio Soybean Council and the SCN Coalition. Since 2018, we have been receiving soil samples from growers to the lab and process those for soybean cyst nematode. We have processed more than 1,000 samples already. And from those samples, more than 65% tested positive for soybean cyst nematode. Fortunately, though, most of those samples that tested positive have low levels of soybean cyst nematode. We do have, however, fields with very high levels. But the point here is that it's really easy, or it's easier, I should say, to keep levels low in a field than to bring those very high levels of soybean cyst nematode down below to a damage threshold. We continue also identifying the different virulence profile of our populations in Ohio. 10% of the population in Ohio, at least from the ones that we put in the greenhouse, are AG type 0 or that race 3. That means that we can effectively manage that population with any source of resistance, including 88788. Unfortunately, though, more than 85% of those populations are that AG type 2, which means that they can reproduce on 88788 above 10% compared to a susceptible. And like I mentioned previously, we have populations in Ohio with a female index of 80% compared to a susceptible. Active management of soybean cyst nematode starts with sampling your field, know your numbers. That's the slogan. Not only know if you have soybean cyst nematode, but how much soybean cyst nematode you have is the same way that you're gonna sample your fields to take a soil sample for nutrients. Break your fields in equal parts, a zigzag pattern collecting multiple subcores. Divide fields in homogeneous soil conditions, or target areas that you have been yielding low. Entryway, areas with high pH, which for some reason allows a nematode to reproduce faster. Send that to a soybean pathology and nematology lab. We continue to process up to 2 samples per grower free of charge. You send that sample, we process for soybean cyst nematode. We return that to your email, your phone number, however you want to receive the results. You will get an email saying non-detected, hopefully, or the amount of soybean cyst nematode X per 100 cc of soil that we have detected on your soil sample. Below, there will be a table with different recommendations according to the level of soybean cyst nematode detected in the field. If you have any questions, of course, feel free to shoot me an email or give me a call. This is a QR code that I prepared and will take you to a folder. Normally, when I give this talk in person, I bring these sampling kits with me and I give it around. It's 2 plastic bags where you can put your soil sample and send it. But within that plastic bag, I print out several fact sheets that really have a lot of information on soybean disease management overall. Soybean cyst nematode, but also fungicide efficacy, fact sheet, seed treatment for different seedling disease, how to scout for Phytophthora and other water molds organisms. So if you use this QR code, it will send you to that folder and you can have access to all that material, including a submission form. That submission form that you can use when you send your soil sample to the lab to test for soybean cyst nematode. I will also have this QR code at the end of the presentation if you don't have the chance to have access to it now. With that, I will want to use the remaining of my time here to talk about the other projects and other pathogens that we are evaluating here in the soybean pathology and nematology lab. So, staying around that rhizosphere of the soybean, let's talk a little bit about other true fungi that can cause problems to soybean, especially in that seedling stage of the plant. So one of the problems that we can encounter early in the seasons are these true fungi. And I say true fungi because the water mold, the oomycetes, Phytophthora and Pythium, they're not true fungi. They're oomycetes. They're a different organism. And why is that important? And I love these pictures from Ann Doran's, where she have these seedlings germinated. Here we have a control with absolutely no pathogen. All the other seedlings, they were exposed to Fusarium graminearum. I'm very glad that peers cover Fusarium graminearum because the same Fusarium graminearum that will affect wheat and also corn can cause seedling disease problems here. So all of these seedlings were exposed to Fusarium graminearum and a fungicide that can control true fungi, Maxim, another fungicide, another active ingredient that can control and manage true fungi, and one that is specific for water mold, for Phytophthora and Pythium, Apron. You can see that when we're using a fungicide meant to control true fungi, we don't see development of symptoms here. On the other hand, when we're using a product that is not meant to control a true fungi, and we have a true fungi here, that organism will kill the plant. Green light. And that takes us to one of the field trials that we had this season, 2022, and that we are going to continue doing in this 2023. So what we do is we grow these different organisms on either sorghum or millet seed, and then we mix it with soybean. And we plant those in our experimental research trials. We infest those plots with the fungus that we're trying to evaluate different treatments. So in this case, we have Fusarium graminearum mixed with soybean, and we plant those on each of these small plots. And we can see the effect of these true fungi. Fusarium graminearum causes seedling disease, which means that we will reduce the stand early in the season. And we can see some stand reduction here. What I'm going to show you is what we did this last season. We evaluated 4 different products in combination. So we evaluated Cruiser Max Vibrance plus Vayanti. Vayanti is a really good product meant to be used for Phytophthora and Pythium, not for true fungi. And then the other treatment we had was Obvious Plus and Poncho, and then Obvious Plus and Poncho plus Relenja, a new product specifically for Fusarium and Rhizoctonia. So what I'm showing you here on the y-axis is the stand count. We go with a meter stick and we evaluate how many plants came out, emergence, on the untreated and inoculated plots. And then with all the other plots with the seeds that received the fungus, but also the seed treatment. And you can see here that as we treated our seeds, you can see the trend of increasing that emergence of the plant. Statistically significant compared to the untreated check, where those Obvious Plus and Poncho, and adding that Relenja. And then for the soybean yield, the same thing. We see that as we treated our seeds, we can see that increase in yield when we have the fungus present, but a seed treatment that can protect soybean health. As much as 14 bushels per acre compared to an untreated seed in a very highly infested plot. With this, I want to remind, of course, everyone that there are other pathogens that will or can cause seedling problems. The water mold in this case, we talk about Phytophthora and Pythium, Phytophthora even being a problem later on. It really requires that seeding water, that those saturated soils for those oospores to produce sporangia, zoospores that will go to the root system and either kill the seed, the seedling, causing dumping off, or eventually adult plants. A lot of species in the field. So if you have a history of water mold, highly recommended to use a seed treatment. Other fungi that can affect the root system is Fusarium virguliforme, in this case causing SDS, sudden death syndrome. The fungus is in the root system. It will produce toxins that eventually will go up and cause the visible symptoms that we see later in the season, around R5, R6, early defoliation and reduction in yield. These symptoms will be more severe in presence of soybean cyst nematode. Therefore, we encourage, if we see a lot of SDS, sudden death syndrome in our field, to take a soil sample and test for soybean cyst nematode as well. With that, I wanna conclude just talking briefly that we are throughout Ohio conducting different fungicide trials. Last season was an extremely healthy season. So we go out there, we rate for frog eye leaf spot in this case, but also Septoria brown spot, also white mold, anything that we can see and we can test our different products. Again, extremely healthy season that we did not detect any statistical difference between products that we evaluated and the checks. This is just one of the many trials that we had. These, of course, highlight the importance of scouting our fields for the presence of the pathogen when we're going to make a decision of applying or not a fungicide, especially in that window R3 to R5 growth stage. And with that, since I only have 40 minutes and it's 40 minutes and 57 seconds, I want to, of course, acknowledge and thank my team members in the soybean pathology and nematology lab and farmer management in the different farm station. Without them, I could not do all of these trials. And of course, our funding sources. With that, I will leave the QR code here that will send you to that folder with the different extension fact sheets if you want so. Thank you.